Hello, everyone, and welcome to this week's edition of the Extracellular Vesicle Club. This is now an event of the International Society for Extracellular Vesicles, or ISO, and we've been running this journal club for almost a year now, since the beginning of the COVID-19 pandemic. Today, I have the pleasure of introducing Blanca Rodriguez. So Blanca is finishing up her PhD at Duke University, and she's going to be speaking with us today about bacterial extracellular vesicles, a topic that we haven't really covered very much in this EV club so far. So Blanca, thank you so much for joining us today. And we're really looking forward to hearing about immunomodulatory properties of bacterial EVs.
Thank you so much for that introduction, Ken. I'm going to go ahead and share my screen. So yes, my name is Blanca Rodriguez, and very excited to have the opportunity today to walk you all through some of the key findings from the paper that we published at the end of last year, which is entitled Staphylococcus aureus secretes immunomodulatory RNA and DNA via membrane vesicles. So I know this group appreciates the fact that we know that all three domains of life are known to secrete these extracellular membrane vesicles. The lab that I work in, we specifically focus on studying membrane vesicle secretion from bacteria cells. And so it's important to note that all bacterial cells that have been studied to date have been shown to produce these membrane vesicles that originate from the membrane of the bacteria. And these vesicles are spherical, nano-sized vesicles that are composed of phospholipid bilayers. They contain insoluble proteins as well as soluble proteins, lipoproteins, as well as other macromolecules. And they range between 30 to 300 nanometers in diameter. And there's been several decades now of research that have investigated and really established the fact that these vesicles are not just being secreted by the bacteria for maybe not just for them to be secreted as waste material, for instance. They're actually quite active, and they have been shown to mediate very important interkingdom and interspecies communication activities. And so in this slide, I just want to briefly introduce you to some of the biological functions that have been attributed to these bacterial vesicles. So these MVs are known to, or they're able to act as decoys against phage and antimicrobial agents. They have also been shown to be involved in the formation and maintenance of biofilms. And because of their structure, they can actually offer protection to their cargo from degradation by proteases or nucleases that can be abundantly found in the extracellular space. And if the vesicles that are being produced are being secreted by bacteria that are pathogens, they've also been shown to deliver virulence factors to recipient cells. And these vesicles have also more recently been identified as novel horizontal gene transfer mechanisms. Essentially, there's plasmids that can be delivered or transferred between different bacterial species through these vesicles. And this is a natural horizontal gene transfer mechanism, which is really cool. There's also a lot of research that has shown that these vesicles can contain pathogen-associated molecular patterns. Again, if the vesicles are being secreted by bacteria that are known to be pathogenic, and these are typically abbreviated as PAMPs. And within this group of PAMPs, RNA and DNA molecules have also sort of come to the forefront as potential modulators of immune response within host recipient cells. So when I started my PhD back in 2015, and then I joined Meta's lab in 2016, I came in as someone who was very interested in RNA in general, based on my background. And so I was excited in the opportunity of incorporating my own excitement for, just RNA biology with extracellular secretion of bacterial EVs. But I noticed that in the literature, unlike what has been, I think, established for decades now, which is that in the eukaryotic EV field, which is that RNA is in fact one of the key components of vesicles, there really was a lag in research and focus on trying to basically accept whether or not RNA molecules could be secreted with bacterial vesicles. And there's a lot of reasons why there was a lot of, and there still kind of is a lot of hesitancy to accept that. And I can discuss that later if anyone's interested. But for now, I just want to make note that there's now about 5 to 6 years worth of research that has actually started to show that, in fact, bacterial vesicles do contain RNA molecules. And so we're starting to appreciate a potential pattern of the type of RNA molecules that get secreted in these vesicles. And so generally speaking, these tend to be relatively short RNA molecules that range between 15 to 200 nucleotides in length. And this includes small RNA, tRNA, ribosomal RNA, as well as messenger RNA fragments. And there was a study in 2018 that actually found that the RNA profile in Salmonella enterica autoimmune vesicles can change according to culture growth conditions. So this was really one of the first studies that suggests that there might be some possible sorting mechanisms for the types of RNA molecules that end up being secreted in vesicles from bacteria. In 2016, there was 2 papers that actually showed that vesicle-associated RNA can be transported inside host cells. And then there was some indirect evidence suggesting that there's potential for some of those specific RNA molecules to affect host immune responses. Now, on the side of the DNA cargo of outer membrane vesicles or bacterial vesicles, there's a little bit that has been known. Other than the horizontal gene transfer mechanisms, there's a couple of studies that have shown that DNA packaged in bacterial vesicles can actually potentially modulate host immune responses. And there was one paper, or maybe 1 or 2 papers, that showed that specifically DNA receptor, toll-like receptor 9, can recognize the vesicle-associated DNA. And then in 2017, there was a paper in Pseudomonas aeruginosa memory vesicles that showed that DNA cargo can actually be delivered into host cells. So there's a lot of questions that are in this specific field. And again, as I was developing this project, there was a few that I really wanted to focus on. Specifically, these are some of the questions that we explored for this paper, which is, are vesicle-associated nucleic acids in fact immunomodulatory, something that I think deserved a lot more attention than it was getting at the time. We were also interested in really figuring out whether nucleic acids associate with vesicles in a way that we would be able to figure out if they're fully encapsulated or maybe surface associated. And the reason why we were really interested in this question is because the localization of the nucleic acids with respect to the vesicle structure is important, especially when you think about how those nucleic acids can interact with receptors inside cells. And then one of the other questions was, what are the mechanisms of vesicle-mediated nucleic acid transmission to host cells? So this schematic kind of just shows the general questions that we were asking and sort of what we were picturing would help guide this project. So as we were developing this project, we were also looking at the literature and decided to study Staphylococcus aureus as our model organism for these questions that we were interested in answering. So Staphylococcus aureus is a gram-positive serious opportunistic human pathogen. It's notorious for developing antibiotic resistance, and it is actually known to be the leading cause of skin and soft tissue infection in the US. And for the purpose of this project, it's also important to mention that Staph aureus has also been established as a producer of vesicles that contain biologically active macromolecules. So there's a lot of work that has shown that Staph aureus not only secretes vesicles from the surface, but these vesicles are quite biologically active and relevant in host pathogen interactions. But again, when I started this project, no one had even looked at the nucleic acid content of Staph aureus EVs to determine if DNA or RNA are found in those vesicles. Additionally, as we were developing this project, we also noticed that there's several studies that have established that Staph aureus itself can produce nucleic acids that are immunostimulatory. Specifically, these nucleic acids from Staph have been shown to interact with receptors that are found inside host immune cells, particularly toll-like receptors that are found in endosomal compartments and some cytoplasmic receptors. And ultimately, a lot of those studies reported a quite potent interferon response. But something that really stood out is that even though we know that those nucleic acids are quite important for host pathogen interactions, there have not been any reports of how bacteria, well, specifically Staph aureus, can secrete RNA and DNA. There's no known secretion systems. And also, we don't actually know how they can be delivered inside a cell to then potentially bind to cognate receptors. So the hypothesis from all of this was just that vesicle production itself could be serving as a secretion mechanism and as a delivery vehicle for Staph aureus RNA and DNA. So in order to begin answering these questions, we first had to establish a method to isolate vesicles, and specifically to account for potential confounding factors that might kind of muddle the interpretation of our data, especially because we're going to be focusing on the RNA and DNA content of vesicles. We noticed that there's been some reports that specific growth media, like LB, for instance, can actually have some contaminating RNA molecules, potentially from like the yeast extract. And so I grew my cells in a media called nutrient broth, which is NB, but I wanted to really be careful and make sure that I was accounting for potential contaminating RNA molecules that were not from bacterial origin. So I actually, every time I prepared my media, I RNased it, and then we autoclaved it, and then we grew the cells in the RNased media. And so doing a growth profile, we noticed that growing our cells in this RNase media didn't affect, at least not drastically, the growth dynamics of the bacteria. So that was good. So everything that I'm going to show you is using vesicles that were isolated specifically from cells grown under RNase to media. And then we're very deliberate in only isolating vesicles from Staph aureus cells that were grown to midlog phase. And this was important because we were interested specifically in looking at, as much as we could control for this, the vesicles and their RNA and DNA cargo that were secreted actively and not just from cell death. So at this growth phase specifically, you see that, and we just demonstrated this using a couple of different viability assays, that at this growth stage, there's very little cell death. So we moved forward and basically focused in on isolating vesicles from the supernatant of cells that had been grown to midlog phase, subjected the supernatant to several filtration and ultracentrifugation steps. And then we ultimately loaded our vesicle samples into an OptiPrep density gradient to further separate the vesicles from any non-vesicular material. And then we always ended up with 6 fractions of equal volume starting from the top. And again, these vesicles are proteolipidic, so we always expected them to sort of float to the top. So as I've mentioned, these vesicles are composed primarily of proteins and lipids. So we normally expect the vesicles to contain high amounts of protein and lipid, and we used different fluorescence-based assays, detection methods to figure out where the vesicles were located in each of these fractions. And as expected, we saw enrichment of proteins and lipids in fractions 1, 2, and 3. We also used some other fluorescence-based methods to figure out if DNA and RNA are in fact present in these vesicles. And this kind of ended up working nicely, where we were able to detect RNA and DNA using those assays, and they also were found enriched in fractions 1, 2, and 3. And then we further confirmed that vesicles were definitely in these fractions by negative stain electron microscopy. This also enabled us to count the number of vesicles that were in each fraction, or at least a representative amount, and to measure the diameter. So these vesicles are generally quite small. I think the diameter was around 65 nanometers. But ultimately, what we show here is that Staph aureus EVs are enriched with RNA and DNA, as well as obviously proteins and lipids. So if you look at the paper, there's a couple of figures that I'm not going to necessarily cover right now, just in the interest of time. But I want to highlight this specific set of data, which basically comes from that question that I mentioned earlier about trying to establish how nucleic acids associate with vesicles. So we initially performed a series of nuclease protection assays, which is, I think, pretty standard for people who study RNA in vesicles. And we did different ways of trying to figure out exactly the susceptibility of those nucleic acids. Ultimately, this was one of the assays that we liked the best because it gave us a lot more sensitivity. And so what we did was we extracted RNA from purified vesicles. And again, consistent with what other groups have reported for other species of bacteria, the RNA that's packaged in vesicles from Staph is very small. It falls below 200 nucleotides in length. And then when we did our nuclease protection assays, essentially the concept is that you have these untreated vesicles that are treated with, in this case, we ended up using benzonase, which cleaves both or degrades both RNA and DNA of both single-stranded and double-stranded nature. And the hypothesis was that upon treating with benzonase, you would remove surface-associated nucleic acids, but you would still have a population that's protected from degradation. So when we were looking specifically just at the RNA from these vesicles, after extracting RNA from vesicles that had been pretreated with benzonase, we actually saw a significant decrease in the amount of RNA that was detected using the bioanalyzer. And you can see that pretty clearly here. So there appears to be some RNA molecules associated with these vesicles that are susceptible to degradation by benzonase, and another population that is not. So then we expected that after treating the vesicles with detergent and then boiling the vesicles, which would essentially just fully permeabilize the vesicular structure, and then following up with treatment with proteinase K in order to disrupt any potential interactions of RNA and proteins, we then also followed that proteinase K treatment with PMSF to inactivate the proteinase K, and then finally we added benzonase. So at this point, after all these treatments, we just really expected the RNA and DNA molecule or the RNA content to be degraded completely. What we saw, and something that we also show with other assays, particularly with denaturing gels and non-denaturing gels, and something that I'm showing you here, is that consistently there is a subpopulation of RNA molecules that are found in our vesicle samples that are extremely resistant to degradation. And this is something that we still kind of haven't quite figured out why that this is the case, but we can at least at this point just make the conclusion that there appears to be two subpopulations. The RNA content of Staph aureus consists of a benzonase sensitive and benzonase resistant subpopulation. Now looking at the DNA content of these vesicles, we just went ahead and did the same thing that I just showed you, but instead of extracting RNA, we extracted DNA. This was also quite surprising, actually. So when we extracted the DNA, we noticed this very faint band that always appeared to migrate around 500 base pairs. And in contrast to what I just showed you about the RNA, even with just the benzonase treatment alone, there was no significant change in the amount of DNA that we could detect. And nothing that we did to the vesicles also changed that. So here we actually found that the DNA content of Staph aureus vesicles mostly consist of benzonase resistant subpopulations. Okay, so we at this point had established that there are RNA and DNA molecules in the vesicles. Some of them might be surface associated, some of them are highly protected and resistant from nuclease degradation. But we wanted to then look at this more interesting question about whether these vesicles are themselves immunomodulatory, and if these purified vesicles can essentially recapitulate the response that other groups have reported for Staph aureus RNA and DNA, specifically in their ability to induce a type 1 interferon response. Now, I'm just going to show a very simplified view of the Staph aureus vesicles, showing like RNA, DNA, and maybe some proteins, but just keep in mind that these vesicles contain a complex mixture of macromolecules. It's not just RNA, DNA, and some proteins that are present. So to start these studies, we decided to work with macrophage raw 264.7 cells, which we grew to confluency, and then we stimulated those macrophages with purified vesicles, and then we measured as a readout for potential RNA and DNA activity, the mRNA expression of type 1 interferons. So right off the bat, we did not detect interferon alpha mRNA expression in response to the vesicles, but we did detect high levels of interferon beta mRNA expression in response to vesicles. And we did a time course analysis, first of all. We found that after 3 hours of co-incubation of our macrophages with our vesicles, that was sort of the peak induction. And we also showed that this was a dose-dependent response. So unless I indicate otherwise, everything that I'm going to show you from now on is going to come from 3 hours of stimulation at 5 micrograms per mil of vesicles. And this is based on total protein concentration. And then I just briefly wanted to mention that we also looked at the ability of vesicles of each fraction from the octopaired density gradient to induce interferon beta. And we showed that fractions 2 and 3, which were the ones that were highly enriched in RNA, DNA, and vesicles, were the only ones that were really active and able to induce interferon beta. So that kind of coincided nicely with the other data that I just showed. So the conclusion from here is that purified Staph aureus vesicles are able to induce significant interferon beta mRNA expression, specifically in mouse macrophages. Okay, so now bringing in some of the data that I showed you about how there's subpopulations of RNA and DNA that are protected from degradation, we wanted to further, this kind of the next logical step is to see how pre-treating of the vesicles with benzonase affects their ability, at least in terms of inducing this interferon beta response. So looking at this graph here, you can pretty clearly see just right off the bat that treating vesicles with benzonase leads to a significant drop in the amount of interferon beta that is induced in macrophages. So there is potentially some RNA and DNA that are susceptible to degradation, and when you remove them with benzonase, you lose some of that activity in the macrophages. But nonetheless, it really stood out to us that these vesicles that were treated with benzonase were still highly active. So they were still highly able to induce interferon beta. So we wanted to further characterize these specific vesicle populations. And most of what I'm going to show you from now on, I'm labeling the benzonase treated vesicles as BMVs. So we had established that interferon beta was induced in response to Staph aureus EVs. And we wanted to look at the specific pathways that are involved in the activation of some signaling cascades that could ultimately lead to this response. So looking a little bit from the top, we wanted to determine whether or not dynamine-dependent endocytosis is necessary for the induction of interferon beta. This is something that has been established for different bacterial EVs that they can enter into host cells through endocytic pathways. And so using Dinosaur, which is an inhibitor that inhibits specifically dynamo-independent endocytosis, we show that when you pre-treat the macrophages and then add the vesicles, there is a significant reduction in the interferon beta response. So that suggests that dynamo-independent endocytosis is one of the major pathways by which vesicles can be taken into cells. So after this endocytic process happens, you then expect the vesicles to be trafficked inside these endosomal compartments. So they are able to localize. And we wanted to confirm whether or not the vesicles specifically and their RNA cargo can be localized into endosomal compartments by doing super resolution microscopy. So we labeled the early endosomal marker or the early endosomes using this early endosomal marker, and that's shown here in red on the macrophages. And then we labeled the vesicle-associated RNA using cytoRNA select stain. And we found that there was moderate co-localization of the vesicle-associated RNA with these early endosomal markers, suggesting that after endocytosis, the vesicles and their RNA cargo, or in some instances, some of the RNA cargo can be localized inside these endosomal compartments. And so we're really excited about this imaging data, but we wanted to follow it up and reinforce our conclusion by using a different labeling method for the RNA, because this, if anyone here is familiar with this stain, it is not specific for bacterial RNA. So you can potentially still have some interference where the stain might somehow also stain some of the RNA from the macrophages. So we wanted to label the RNA specifically as it's being synthesized in the staph cells, and we were able to use this bioorthogonal click chemistry method, which utilizes 5-ethanoyuridine. So you just grow your Staph aureus cells in the presence of this uracil analog. You grow it to mid-log phase. Again, that's where we've been growing all of our samples. And then you expect the newly synthesized RNA to incorporate this 5-ethanoyuridine. And so we initially just wanted to do a proof of concept that using this method, which has been done for other bacterial species, but had not been, at least not that we could tell, published for Staph aureus. We just wanted to make sure that we had the right parameters to label the RNA in staph cells. So we permeabilize the cells after attaching them to polylysine cover slips. And then the click chemistry reaction is copper catalyzed, and essentially the alkyne group of the 5-ethanoyuridine clicks with the azide group that is attached to a dye. Specifically here, we have the Alexafluor 48 dye. And so that enables you to, after permeabilization of your cells, to very specifically label the RNA in your cells. And so we did, again, just a proof of concept where we imaged staph cells and confirmed that, as shown in green, we were able to use this method to label the RNA inside these cells. So then we went ahead and incorporated this into our vesicle isolation method. So we grew our cells in the presence of the 5-ethanoyuridine, and then we isolated vesicles from this culture. And so at this point, we expect any of the vesicle-associated RNA to have incorporated this 5-ethanoyuridine. We furthermore also labeled the vesicles themselves using this lipophilic DID dye. And so we expect the vesicles to contain the dye on the surface of the membrane or just in the membrane, and then the RNA is supposed to be labeled metabolically. So then we did our co-incubation step with these vesicles that had been pre-labeled, grew them for 1 hour together, and then we washed unbound vesicles, permeabilized and fixed the macrophages, and then we did the click chemistry reaction to label that RNA from bacterial origin. And then we imaged these macrophages using Aeriscan super resolution microscopy. And so we ended up getting some really nice images that further just reinforce what we saw previously. Although here we're not doing the co-localization with the endosomal marker. We're just looking at the vesicles and their RNA cargo. So the vesicles are shown in magenta, and then the metabolically labeled RNA is shown in green, and then the DAPI is for the macrophage DNA. So after an hour, we just show that the Staph aureus vesicles and their associated RNA cargo can be delivered into wild-type macrophages. Okay, so we've established that vesicles can get into cells through endocytic pathways. They can localize into endosomes, and their RNA can also localize into endosomal compartments. So then we wanted to further look at something that's really important about endosomal biology, which is this important maturation step that is driven by the acidification of the endosomal space. And the reason why this is important is because at this point we were hypothesizing that the vesicles, because they localize into endosomal compartments, And it is known that within these endosomal compartments, toll-like receptors that are known to specifically recognize RNA and DNA from bacteria, for instance, can then lead to a signaling cascade that leads to this interferon beta response. And endosomal acidification is actually necessary for these toll-like receptors to become active after they recognize the DNA and RNA ligands. So we were able to use baflomycin and chloroquine, which are two inhibitors of endosomal acidification, to determine whether this endosomal acidification step is necessary for the response that we had been measuring. And so we tried baflomycin by itself, chloroquine by itself, and then in combination. And you can see pretty clearly that all of these inhibitors, both of these inhibitors by themselves were able to significantly reduce the amount of interferon beta that was produced in response to vesicles, but together they were really efficient in really abrogating that response. And so that, to us, suggested that endosomal maturation contributes significantly to the vesicle-mediated interferon beta induction. Okay, so at this point, we have shown, again, endocytosis, localization inside endosomal compartments, interferon beta response, and we started to put together the pieces of what is known about toll-like receptor signaling, which is that toll-like receptors that are able to recognize nucleic acids, they signal downstream to two specific adapter proteins. One of them is called TRIF. This is this functions downstream of TLR3, and then MyD88, which is necessary for the other toll-like receptors to signal. So we were able to get our hands on bone marrow-derived macrophages that did not express these two adapter proteins, TRIF and MyD88. And then we compared the response of interferon beta in the double knockout cells to the wild-type macrophages. And it's pretty clear here that these two proteins are quite necessary for the induction of interferon beta, because you see very little to no response of those cells when you don't have these two adapter proteins. And then we looked even further downstream, and we looked at IRF3, which is a interferon regulatory factor, and IRF7. So we were able to get these macrophages, and we also found that there was no activity when macrophages do not express these two regulatory factors, which are known to also function downstream of these TLRs that lead to the interferon beta response. So then the next logical step was to knock out specific toll-like receptors of interest. So toll-like receptor 3 is known to recognize double-stranded RNA and tRNA. TLR7 can recognize single-stranded RNA as well as some tRNA molecules. And then TLR9 can recognize CpG DNA from bacteria. So in all cases, macrophages that did not express these specific toll-like receptors had exhibited very significant reduction in their ability to induce interferon beta. And so the conclusion from this was that endosomal nucleic acid receptors are activated in mouse macrophages specifically upon vesicle exposure, and this largely occurs through endosomal RNA and TLR and DNA receptor signaling. Okay, so lastly, because we had noticed that very interesting aspect of vesicles in terms of the fact that there are some populations of RNA and DNA that are highly protected and resistant to nuclease degradation, we wanted to look at the potential functional consequences of that. So again, we just, using the macrophage background cell line that we just used with all the knockout macrophages, we just wanted to confirm what we saw previously with the Rod264 cells, which is that treating macrophages with benzonase-treated vesicles leads to a reduction in the amount of interferon beta. So this is not really something new. This is what we expected. It's just in a slightly different macrophage cell line than what I showed up top. But then we wanted to see what happens when we then treated the TLR3 deficient macrophages, for instance, with vesicles that were not treated with benzonase. Again, mostly everything that I've shown you up to this point is using vesicles that were pre-treated with benzonase. So when we added these intact vesicles to the TLR3 minus cells, we saw a significant amount of more signal in terms of the interferon beta expression. So that actually suggests that some of those RNA molecules that are, or DNA molecules that are removed with the benzonase treatment, that they can potentially signal through other receptors that are not TLR3, or there might actually also be some TLR3 that's active there. But because of that signal difference, that's sort of the hypothesis. Now, what was really interesting is that when we did this same experiment with the macrophages that did not express TLR7 and 9, we saw no significant difference. So those nuclease sensitive RNA and DNA molecules didn't really change how much interferon beta was expressed. And so combined, these data suggest that there is a population of vesicle-associated RNA or DNA that is exposed and that can enter inside macrophages and signal through intracellular receptors that might include TLR7 and TLR9. So with that, I wanted to here to summarize some of our conclusions and what we have established with our paper, which is that Staph aureus, well, we essentially show for the first time that in cultured mouse macrophages, interferon beta mRNA induction can be triggered just by the purified Staph aureus vesicles. We also show that vesicle-induced interferon beta expression requires endosomal TLR activation, and this is specifically promoted by the engagement of these endosomal toll-like receptor 3, 7, and 9, and this points to the likelihood that vesicle-associated and protected RNA and DNA molecules are inducing this response. And we also note that nuclease protection assays revealed that there's subpopulations of nucleic acids that are protected from nuclease degradation. Maybe some of the reason why there's certain populations of RNA that are highly protected could be due to secondary structures or non-proteinaceous binding partners that are protecting the nuclease-resistant RNA from degradation. And I do want to mention that although we were unable to determine the exact nature of the association between DNA and vesicles, based on the work that we did with the TLR9 deficient cells, we do think that there might still be some nuclease-sensitive DNA that are in these vesicle samples that we just were not able to capture with the assays that we did for the nuclease protection assays. So there's more follow-up that needs to be done. And actually, I've done some follow-up with some immunogold labeling of double-stranded DNA, and I do see some surface-associated DNA. So it's a little bit of a teaser for a potential follow-up to this work. So there's a lot of work that still needs to be done to further reinforce our findings and to ask some questions that are outstanding. So one of the things that I mentioned and I focused on a little bit was these endosomal acidification, which we know is necessary for TLR3 activation. But another hypothesis that we have that is not mutually exclusive to what is known about the TLR activation is that there is potential for the conditions that the endosomes have during the acidification process that might lead to the disruption of the vesicular structure. And if that happens, then there's potential that the nucleic acid content could be released and that could enable it to be recognized by the cognate receptors. So that's something that we're trying to investigate a little bit more, whether a low pH condition, for instance, could lead to changes in the vesicle structure. We are also in the process of doing RNA-seq on these vesicles, finally. And some of the optimization that I'm going through right now is trying to get enough RNA to be able to generate cDNA libraries. So if anyone has any advice or suggestions, I would highly appreciate that. Something that I also kind of want to investigate a little bit more deeply before I graduate this summer is whether there's other vesicle uptake mechanisms that are involved in this response, since I was really only able to study the effect of dinosaur. And then there's also future studies that will be necessary to address whether the interferon beta induction of Staph aureus vesicles has an impact on the pathogenesis of staphylococcal disease. In terms of host pathogen interactions, it is important to establish whether this very potent response is beneficial for the pathogen or if it's beneficial for the host cell to potentially clear the pathogen. So with that, I just want to give a shout out to my lab, specifically to Maida Keen, who's been highly supportive and has really allowed me to explore questions that had not been previously explored in her lab. My awesome lab mates, some of which have graduated by now, some of the facilities and individuals who have provided reagents and advice, and some of the funding agencies that have also allowed this project and this paper specifically to be published. And if there's any questions, I would be happy to chat either through email or on my Twitter. So with that, I am happy to take any questions, and thank you so much for being here and for giving me your attention.
And thank you for this presentation. I really enjoyed this, and I'm really glad to see how you were being very careful about dissecting these different ways of presenting the nucleic acids to the receptors. So that was really encouraging to see. We have quite a few questions that have come in already, and I just remind everybody, if you do have questions for Blanca, please place them in the chat box. But I want to start with a couple of questions here that were from Phil Askinase, who asks, well, first of all, he asks about the function of microRNAs in bacterial vesicles. So are there bacterial microRNAs, first of all?
So the actual, microRNA, in bacteria, it's called small RNAs, and they're a lot larger or longer than the microRNAs, which are, I believe, maybe up to 35 nucleotides in length. In bacteria, the small RNAs are between, I want to say, maybe 20 to 200 nucleotides in length. So they're actually a lot longer. I'm not familiar with microRNAs specifically that are known to, well, I guess the small RNAs are analogous to microRNAs because they are known to regulate the expression of other messenger RNAs within the bacteria cell, specifically in response to changing environmental conditions and infections. So that would be the analogous, but there's actually a lot less that is known about bacterial small RNAs relative to microRNAs. And you don't have like the dicer proteins, for instance. You do have some similar proteins that are sort of shuttling some of the RNAs and acting as chaperones, but yeah, there's a lot more that needs to be done in this area, I think, at this point. Hopefully that answers the question.
Yeah, I think so. I think it's important to think about the distinctions between the bacterial mechanisms for making these small RNAs versus what we see in some other organisms with specific processing of the microRNAs that end up being quite a bit shorter. So, but what about this? This also raises a question for me. What about delivery? Do you think there could be any delivery of the bacterial RNA into the cytoplasm of a recipient host cell?
Yeah, it's also a really great question. And one of the papers that I mentioned in the introduction actually specifically focused, after their deep sequencing of the RNA, specifically from Pseudomonas OMVs, they found that there was one small RNA that was highly enriched in the vesicles. And they kind of did a little bit indirect because they did transfection of that specific synthesized RNA. But when they did the transfection, the idea for them and the ultimate conclusion was that that small RNA could somehow interact with the host messenger RNA. So it's doing this like inter-kingdom RNA interference type of mechanism, which is super fascinating and cool. But I think that there needs to be more investigation into whether that's something that vesicles can also mediate, because transfection is different really than the vesicles. But there are some studies that suggest that membrane fusion between the vesicles and host cells could happen, and that could be one pathway by which you could have just a direct dump of the RNA or other cargo into the cytoplasm. So there are some indications in the literature that that could be happening. I just think there needs to be more to fully establish that. But it's super fascinating, I think, to think that that would be an option as well.
Great, thank you.
Mm-hmm.
So I'm going to continue going through the questions, and I'd just like to let everyone know that I am allowing unmuting now. So I will ask the question, but if you want to jump in and follow up on your question, please do so. So I'm going to go next to someone, I'm not sure who this is, PPNDL2, who asks, how do you separate bacterial EVs from bacterial spores?
Bacterial spores.
Or is this a problem in your system?
Yeah, so this is not a problem with Staph. I think there's definitely like a Bacillus subtilis that is a spore-forming bacteria. I'm not super familiar, but I suspect that the spores are larger in size, and so you end up filtering those out. But yeah, it's not something that we have encountered with Staph aureus, fortunately.
Great, okay. We have a question then from Fanuel, whose mic is also not working. But when cells are treated with your membrane vesicles, do you think there's a possibility that the response could be related to the proteins? Or interaction of proteins and nucleic acids. So did you do any characterization of protein content?
Yes, I actually have like I was going to include this and then it was already a little long, so I didn't, and it is in the paper. So there was a question when we were going through the review process about potentially specifically, one of the important proteins that are found in Staph aureus is this protein A, staphylococcal protein A SPA. And there's a paper or two that have actually shown that SPA from Staph aureus can induce interferon beta responses in host recipient cells. The mechanism of how that happens is not known, but it has been reported that this SPA protein could be also, and it's also been shown to be in the proteome of vesicles from Staph. So we did do those experiments where we try to figure out if this protein A from Staph aureus in vesicles is also contributing to the effect. So we were fortunate that one of the labs that works across from us is actually studying protein A. And so they have a Newman Delta SPA strain that we were able to use. We use Staph aureus Newman, so this was a perfect match for us. But we just grew the cells the same way that we did the wild type cells, isolated vesicles, which you can see that they look vesicular, and maybe there's some follow-up that I could do to see how they differ from the wild type vesicles, but I haven't done that yet. But we did do the functional experiments where we looked at the interferon beta response from wild type vesicles versus the deletion of this specific protein. And so there does appear to be a significant reduction in activity levels. So we think that protein A might be contributing to the response. But when we also looked at the protein profile of vesicles from this Delta SPA mutant, the protein profile and also the proteinase K assays were widely different from wild type. So we are not sure. We're kind of careful with concluding that it's protein A specifically that is leading to this reduction. And maybe protein A is also affecting the biogenesis of vesicles or the cargo selection and sorting, because it is a very important protein that's actually bound on the cell wall of Staph. So we think that it might be contributing, but we also think that there needs to be more work that needs to be done to determine whether or not protein A itself is an important protein for biogenesis and sorting of cargo. Hopefully that explains. That's kind of the one thing that we did look at. And I actually also recently did some proteomics on my vesicles, and there's some follow-up that we need to do to sort through that data. But yeah, I think it'll be good to look at potential candidates that could be contributing to this response. Thank you for that question.
We have several questions from Xiaogang Wang. So Xiaogang, would you mind please unmuting yourself and you can ask those questions, because I see you have a few.
Hi, it's really nice talk. I have a couple questions.
Sure.
Yeah, so the first question is about your immunofluorescence imaging experiment. So you showed the entry, the EVs, and see they are localized at the endosomal compartment. So I want to know how long you incubate the cells with EVs. Did you do a time course to look at whether there are some changes for their cellular localization?
Yeah, that's also something that I want to explore a little bit more as part of the uptake mechanisms. In this instance, I did a 1-hour incubation. So this was, I wanted to just make sure that I was doing, it was a little bit arbitrary, to be honest, because all of my activity assays were after the 3-hour incubation. So I wanted to make sure that I was looking at the uptake of the vesicles and their cargo sometime before the 3 hours. So 1 hour just was kind of convenient. I've also done experiments with 30 minutes. And actually the first figure that I showed, not this one, but this one here, this was after just 10 minutes, the localization. So I think that there's populations of vesicles that are able to be taken into cells rather quickly, because this was, I believe, either 10 or 30 minutes. I should have labeled this a little bit better to remember. But I have done 10 minutes, 30 minutes, and then 1 hour, and then maybe an hour and a half as well. Not everything made it to the paper, but I think I need to do more controls to really look at, the patterns and to see if any of this ends up being in the cytoplasm at all. So, yeah.
Okay, so I guess what I'm thinking is, if you incubate the cells with EVs longer, it's possible they can traffic to some other cellular compartment, not just in the endosome. Because in the endosome, it's kind of like, if they endocytosed, this kind of the first step, right, after they enter the cells. So they might, after that, gonna, traffic to other place or, degrade or whatever, by the cells.
Yeah, absolutely. I think that's for sure one of the things that needs more follow-up is to really look at what happens over time. And I would be interested in doing longer time points. Actually, something like 24 hours, I think would be pretty feasible for me to do, to see what happens relative to these super early time points, to see if, yeah, if there's just completely, degraded in the lysosome at that point, or if somehow some of this content ended up being released and somehow found its way into cytoplasm or nucleus, which is something that a group reported for DNA from vesicles, which is, and I think RNA as well, which is really fascinating.
Yeah. So, and I have a couple more questions. One is, when you did the inhibit treatment experiment, one is with dinosaur. So you mentioned if you pre-block the cells with dinosaur, can block the type 1 interferon response, right? So I think the point that you mentioned, we should be more careful for that, because I think a tolerant receptor signaling, the endosomal TLR signaling is really dependent on their endosomal trafficking. So if you treat the cells with dinosaur, then induce the tolerant receptor with any of tolerant receptor ligand, probably the type 1 interferon response also can be reduced. So in that case, for the experiment, if you treat it with dinosaur, can block EV-induced type 1 interferon. It doesn't mean this depends on the EV entry. So this is one point. Another point is you also, like if you pre-treat cells with BAFA, the inhibit to block the acidification, right? So there's, I think, a pretty well recognized. So the acidification of endosomal is really required for the interaction between tolerant receptor and the nucleic acid. That means that's important for the type 1 interferon response activation. So in that case, if you treat the cells with this inhibitor BFA or other, acidification inhibitors, so if the EV-induced type 1 interferon response is induced, but that probably doesn't mean the nucleic acids are released from UVC at the endosomal.
Yeah, those are really important caveats for sure. And because dinosaur and bafomycin and these inhibitors are more like broad range, I definitely agree that we do have to be a little more careful. I know that was one of the things we got in the review, and I still sometimes when I present forget to mention that caveat. So I'm glad that you brought that up, that dinosaur, while it is known to block uptake, it still has other sort of non-specific effects, which, like you mentioned, signaling of TLR9 can be disrupted just by the dinosaur treatment.
Exactly.
So it requires definitely a more careful analysis. One of the things that I mentioned with what I want to follow up with, the uptake mechanisms, is doing labeling of, imaging. What happens after we do dinosaur? Can you see that there is a reduction in the uptake? That is a little bit more indirect than what I am showing here in a way, because it's not going to necessarily tell you whether the interferon itself is also reduced, but I think it'll start drawing that picture. And I know that the paper that I think your lab published showed that dinosaur does lead to a blockage of uptake. So I do need to follow up a little bit more on that. So I appreciate those comments for sure.
I think if you have some positive controls, which means like some tolerant receptor ligand, if you include that in your experiment, it might be can tell more, give you more answers about that.
Yeah, finding the controls is going to be, and I think there's some stuff that I could look into. I know for other uptake inhibitors, there's already some pretty well established controls, but I need to look a little bit closer for dinosaur, for instance. Thank you.
Yeah, thanks.
Great, very good. So I'm going to skip down to Genevieve asks about receptor-mediated uptake. So are surface proteins important for taking up these bacterial MVs?
So many great questions today. That's a really good point. So that's something that we can't rule out. I'm trying to remember if there's examples of Staph aureus EV, if it's known yet whether specific receptors are known to bind to Staph EVs, because there are, like I mentioned, a variety of different proteins on the surface of vesicles. It's not just, the nucleic acids. So, and there's lipoproteins as well that are known to specifically interact with a toll-like receptor 2, which is localized on the surface of cells, immune cells. So that would be potentially one way that you could get an interaction and that could lead to
I'm sorry, I didn't get which receptor.
Toll-like receptor 2, I think, is known to interact with lipoproteins from bacteria, and Staph aureus is one of those. But I can't comment super confidently about that because off the top of my head, I don't know if it's been reported for EVs. But oh, go ahead.
No, I was just curious. I thought I may have missed it. I'm very curious about interaction between bacteria and eukaryotic cells. So I was wondering if it was known that some specific protein on the surface or the nucleic acid might interact with non-receptor.
Yeah, there's also other receptors that are surface associated. There's one called RAGE. I don't remember exactly what it stands for, RAGE. But I remember when I first started working on this project, there's a paper that stood out that shows that DNA can bind to a surface protein in mammalian cells, and that can lead to like endocytosis of that DNA. I don't remember the details of all what happened after that, but there is some known DNA binding receptors on the surface of cells. But there's also, like I mentioned, proteins on vesicles and other bacteria. You know, if you're looking at gram-negative, they have LPS, and that binds with TLR4, which is also surface associated.
Well, just one follow-up on this. I was wondering about if these MVs are more rich in glycan or than lipids, or are they really that similar to eukaryotic EVs?
Yeah, that's a great comment as well, because you know, with Staph aureus or gram-positive bacteria, there is a cell wall that comes after the membrane. So there's investigation into how the vesicles are even released from these organisms, and there might be an opportunity for certain fragments of the cell wall to somehow still be associated with the vesicles. Either, I'm not complete speculation because we don't know yet. So we don't know if these vesicles are just entirely composed of just membrane, like lipids, or if they also contain fragments or parts of the cell wall as they transverse that cell wall. There's indication that some vesicles, even from gram-negative, have peptidoglycan components, and they can activate responses inside because peptidoglycan is a very bioactive immunomodulatory molecule as well. So active area of investigation, but we don't fully know yet.
Okay, thanks a lot.
And I think you've you simultaneously answered James Drummond's question as well about that.
Didn't see that question.
Very good. Okay. James, did you have a follow-up?
Well, I just wanted to get at whether you had any more information on how these things get out of these bacteria, especially being a gram-positive. It seems it would be more problematic, but I guess they have solved it.
Yeah, so there's a couple of great papers that were published by Jean Lee's lab on Staph aureus vesicle biogenesis, which is really the only indication we have so far that during specific times of the cell cycle of Staph aureus, there are some autolysins, and these are enzymes that are particularly important for cell division to be initiated. And these autolysins are known to degrade parts of the cell wall of the peptidoglycan. So it's believed that in combination with some phenyl-soluble modulants, which basically act as surfactants on the membrane level, you start to get budding off of the membrane in form of vesicles, specifically in areas where there's also autolysins that are sort of chopping up regions of the peptidoglycan. So you can kind of think about little paths that are being formed at that same level. There's vesicles that are being released. Whether that's genetically regulated, we don't know yet, or whether that's just because of turgid pressure that is building up inside the cell when peptidoglycan is being maybe chopped up at different stages. I think I'm kind of just speculating and taking from those really great studies, but also that's even when you do knockouts, vesicles are still being released, and we're still trying to figure out exactly how this can happen in organisms like gram-positive where there's that thick cell wall.
Okay, thank you.
No problem. Thank you.
So speaking of Jean Lee, there's a question, I think, from two people now, that for which strain did you use?
Yeah, so I used the Newman strain, which is a laboratory methicillin sensitive strain. And I didn't use, I think if I'm also looking at the follow-up of did I just use one strain or others, we only used the Newman strain. I think in hindsight, we have other strains in the lab, like MRSA strains, that would have been nice for us to also compare their activity. The only thing that I did early on in my project was a little bit too ambitious, was I was trying to compare the activity of vesicles from Staph to a probiotic called lactobacillus. So that was the only sort of difference. That didn't make it into any of this, any of the paper, but yeah, I think that's something that needs to be followed up on different strains and maybe more clinical relevant strains as well.
Now, what about the isolation method? So we have a question of, would your method work for gram-negative bacteria?
Yeah, absolutely. So this method actually has been one of the pioneers in the field of outer vesicle research, and her lab has established a lot of the protocols that are now widely used. So I essentially adapted a lot of the methods that we already used in the lab and methods that were reported for Staph EV specifically. There's a lot of variables. A lot of people actually end up isolating vesicles from cells that are grown overnight, so stationary phase, because you do end up getting more of a vesicle yield. But again, because of the nature of the project we were doing, we ended up going with like mid-log phase. But yeah, I mean, everything is pretty straightforward, where you just pellet your cells very gently. You don't need any like big equipment at this stage just to separate the vesicles or the supernatant from the cells. I used two different filters, 0.45 microns the first time around. And then before I do the hard spin here, I do a 0.22 micron. And I actually didn't include another step that I do here, which is tangential flow, because I do start off with normally 3 liters. I've gotten up to 9 liters of starting culture conditions, and that's very time intensive and laborious, but sometimes you kind of have to go as a graduate student through those growing pains. And at that point, using the tangential flow allows you to concentrate the supernatant into work volumes that are more amenable for a graduate student to work with. Sorry, my mouse is going a little crazy. So, and I think a lot of the works that I've seen with mammalian EVs and exosomes end up using very similar processes in terms of the OptiPrep gradient. So hopefully that's something that people are familiar with.
Great. Yeah. And I think you also just answered a question about the filters. So we had one of those.
Mm-hmm.
May Mahoney asks, do you know whether exfoliative toxins are on the surface or encapsulated in your vesicles?
Oh. Exfoliative toxins.
So can I explain a little bit?
Yeah, that would be great.
Please do, yes.
Okay. So we in the skin field, we're interested in diseases such as bullous impetigo and scalded skin syndrome. And in bullous impetigo, you see localized lesions, and then all of a sudden you might get this massive generalized lesions in scalded skin syndrome. And basically the Staph aureus bacteria will secrete a toxin called exfoliative toxin, and it cleaves adhesion proteins on the outside. It's a serine proteinase. And the question has always been, how are these toxins be so at a high concentration that it can target these adhesion proteins? And then the second is, how does it go from localized to generalized? So I've always wondered whether or not these bacteria would then induce a massive release of vesicles that carry these toxins either on the surface or protected on the surface or within the vesicles that would then produce that generalized, disease that you see. So I just thought,
yeah, that's really fascinating and important to look into. So one of the maybe limitations of our work is because we are just looking at the Staph that's grown to midlog phase. I'm not sure if all the toxins are expressed yet at this point. because we did recently get some proteomics data back from what is found in these vesicles relative to the whole cell. I did notice that there was not as many toxins as I would have expected, as other groups have reported for Staph aureus EVs, and those groups have looked at EVs from, grown in different conditions for different amounts of time. So I would have to look a little bit more at my data, and I am really fascinated by the fact that that's something that, because when you are looking at a very potent effect from, like you said, you kind of need a release that's coming from the bacteria. Vesicles are, I think, time and time again showing to be the mediators of a lot of that. But I am not familiar with that on my end. I think it's something that would be really interesting to look a little bit more at, for sure.
Leaf Anderson asks, have you looked to see if virulence factors like alpha hemolysin are carried on the EVs or the MVs? And then we also have a question about complement. So does complement bind MVs or enhance their uptake? Do you know that? But you in any case have not looked at the.
We have not. No, we have not. But that's actually an interesting question, if that could enhance uptake, actually, because I think that's worth looking into for sure.
Just to elaborate a little bit. So, hi, Mallory. Were your, I guess I should have asked this more directly, were your experiments done in the presence of serum or in the absence of serum? So serum might contain the complement factors, whereas, if you did it serum free. So were any of those serum factors required for the uptake of your EVs or MVs?
Right. Oh, that's a great question, because I just use the same. So yeah, I used FBS in my media, which does contain serum. I never used serum depleted media, so I wouldn't know from the data that I collected whether that was important. But I think that would be a very good and easy, relatively easy kind of experiment to do, to check what happens when you deplete it of the serum. Mm-hmm.
Yeah, very interesting question. Thank you, Mallory. So I'm afraid that we're going to have to end our session here today for now. I apologize to those of you whose questions I had to skip over or didn't get to, but please feel free to reach out to Blanca as well. I'm sure she'd be happy to take your questions offline. And so I'd just like to, again, thank Blanca for presenting to us today. I really enjoyed this presentation. I think we had a good discussion as well. And thank you to all the participants for joining in the ICEB EV Club today. We look forward to seeing you all again soon. Thank you.
Thank you.